My name is Daniel Heller. I'm a bioengineer at Memorial Sloan Kettering Cancer Center. I was a teacher before I was trained as a biomedical engineer. I taught 7th and 8th grade physical and life sciences, and that got me really inspired to actually do science and become a scientist. Many people have developed new medicines to target mutations in cancer. These drugs are very effective, but they have serious side effects, and it would be a game changer if we can get these drugs to the sites of the disease and avoid the healthy tissues. My job is to find ways to make these drugs more effective. We make these particles that are filled with drugs, and the outer surface of these particles are sticky, and we can get these particles to go where we want them to go because we can make that surface into something that sticks to the tumor. What we've created is something like a homing device for tumors by making a particle that homes in on certain markers in tumor tissue. And also, they're filled with precision drugs that target certain mutations in tumors, like RAS mutations. RAS mutations are causes of many types of cancer. The tumors that are driven by RAS mutations are often very hard to treat. The precision drugs that are good for targeting RAS mutations have specific side effects that make them intolerable when they're given at a certain amount. Unfortunately, the doctors usually need to give an amount higher than that to cause the tumor to go away. And if we can steer the drug away from certain tissues in the body, we can improve the drug effectiveness much more, but also not make the patient feel so sick because of the drug. It was discovered by other researchers that there are processes like inflammation that cause tumors to grow and to metastasize. And so our innovation was that we started to use this as a targeting mechanism for drugs. We're homing in on the response of the body to the tumor to get drugs to get into the tumor. So we're kind of hacking the process of the tumor metastasis to actually target the drugs to the tumor. If we figure this out, I think we can apply this to many types of cancers because we can put almost any class of drug inside of these particles, and those drugs can be targeted to different mutations, and they could really be effective against many types of cancer. The Pershing Sohn Prize is going to enable us to do a lot of really innovative work that we wouldn't have been able to do otherwise. Living in this time when we can make these precision drugs is really exciting, but I think if we can get them to the tumor and prevent their side effects, we can make them much more effective, and I think that's even more exciting.